talking to you about a relatively recent line of research that I have been engaged with, and it relates to leveraging microbiomes and the information that they contain in the context of respiratory medicine. I will show you a few examples. So I'll start with just setting the scene in brief introduction, and then I will show you several applications of the methodologies and techniques that we have been exploring and developing in the past few years in this space. So starting from the big picture, I think it's very well established now that the human microbiome is bringing a real shift in terms of applications for translational research and clinical care. And it has been attracting immense attention over the past maybe 10 or so years. And the number of research reports and studies are growing really exponentially. It's also kind of been known for a long time that humans have evolved alongside their microbiomes. And these are really living in a symbiotic host microbe relationships, which have a lot of positive as well as negative implications in terms of health and disease. Microbes are essential for synthesis of hormones and vitamins, digestions, and kind of utilization of micronutrients. They modulate our immune system, and at the same that links it to the involvement in various, not just this, other things as well, disease conditions. So there are some representative examples here across the board. I need to point out that in this talk we are looking, in addition to bacteria, at other microbiomes as well, and that's been the kind of the main idea behind this work is combining and hence integrative microbiomics. So in the context of respiratory disease, microbiomes are implicated in a variety of conditions such as asthma, COPD, bronchiectasis, cystic fibrosis, and idiopathic pulmonary fibrosis as well, amongst others. In today's talk, I will predominantly show examples looking at bronchiectasis and COPD, but I do believe that some of these techniques and some of these results could also inform studies on other respiratory disease and conditions. So as I already mentioned, our aim has been to leverage this information. However, we quickly realized that there are a number of challenges that need to be faced and overcome in order to successfully do that. Most studies, I also mentioned this briefly, most studies to date have analyzed microbiomes in singular, in isolation, looking at just at the bacteria or the fungal or the virome, for example. This basically does not take into account the fact that these microbial communities coexist and interact between each other. So this is one of the things that we aimed to look at and to take into account, and hopefully I'll show you how we did it. So our aim was to build a computational framework to integrate bacterial, fungal, viral, and other data sets that are derived from patient populations. And we named, or we use the term of, to define this process, we use the term integrative microbiomics. So this is a slide here summarizing the main data sets and approaches that I will show you today in this talk. And it's a picture of our student giant, whose work I'm presenting to you today. He's jointly supervised by myself and my clinical collaborator on NTU Exeter, NTU in Singapore, and Exeter joint PhD program. So this is a picture from a review that Jan wrote last year. Basically, here we're looking at data that is obtained either by targeted sequencing or a metagenomic sequencing approach. And I have a slide to explain this a little bit more, and maybe some of you in the audience are very familiar with this. So this results in very huge data sets that come very high dimensional, that come with properties which need to be taken into account in the analysis, such as compositionality. They're very sparse, and there may be also batch effects that need to be taken into account. From the computational point of view, and where we are focusing predominantly with Jan's projects, is to develop the computational technique that can be successfully applied for the purposes of answering clinical questions. So we have looked at developing integrative analysis techniques. Jan has tried to apply machine learning and is working on this further. We have looked at microbial association analysis, topological data analysis, and began to try to start modeling mathematically, mechanistically, the interactions between microbial species and data sets. So I'm going to show you examples of some of these, starting from the integrative analysis and microbial association analysis. As a case study, I'm going to use a data set obtained from patients with bronchiectasis. Again, some of you in the audience may be very familiar with this condition. I will briefly introduce it. So bronchiectasis is a chronic lung disease. It's characterized by permanent and irreversible dilation of the bronchi. The main clinical manifestation is productive cough, airway obstruction, and recurrent infection. There's a kind of this view of the vicious circle that underlines worsening of this condition. Basically, during exacerbation, which is worsening of the patient conditions, there is an accumulation of mucus in the lungs, which then increases the number of microbes that can live there and colonize it, interact. That leads to infection and further damage on the airways, which then in turn allows for more mucus to be accumulated and more microbes and infections to happen. So there is unfortunately, eventually patients go through periods of worsening of this condition that are more and more severe. And currently there is no proper cure for this condition. Usually these conditions are managed by antibiotics and combinations of antibiotics. So there's basically a need clinical need and for understanding and providing new ideas in terms of treatment for these patients. In addition to all this, bronchiectasis it's well established that it is a heterogeneous disease that makes it very challenging to interpret clinical trial data. So one of the questions that we wanted to address is, can we stratify patients based on their microbiomes? Is this a strategy that we can take, and can this be useful in terms of clinical translation? Before we started this project, it was established, or there was some studies reporting on association between microbiomes in isolation and clinical outcomes. However, these were all based on single kingdom models, and used the dominant bacterial taxa. Prior bacterial microbiome studies actually illustrated minimal actual change during exacerbation. In other words, looking at whether the dominant bacteria or fungal or other microbial composition in the lungs of patients before and after, or this worsening of their condition known as exacerbation, if there is a change, there was no evidence that this could be demonstrated robustly. So we hypothesize that we will be able to achieve patient stratification if we integrate the information by different microbial species and kingdoms extracted from patient samples. So that was our beginning starting point in this project, in this investigation. I did mention that the data comes from two main approaches. One of them is the targeted sequencing approach, where a specific region on the DNA of the microbial species, bacteria or fungi or others, is used to identify what type of species are present in a sample. So basically that gives an information on who is there. Metagenomics is a whole genome sequencing approach. It's relatively newer. It's newer compared to targeted. It's more challenging in terms of extracting information. But in addition to answering the question, who is there, identifying species, or taxa, it can also it also provides information about what they might be doing and what kind of signaling pathways these microbes may be involved with. So in our work, we have used both data sets derived using both of these approaches, and I will show in a minute how with the example of bronchiectasis how we have done that. So in the case of the bronchiectasis patients cohort, we had 217 patients. For each sample, the so-called bacteriome or the bacterial composition, the microbiome, the fungal, and the virome were available. These are patients from several different hospitals, actually not just in Singapore, but also in the UK from Dundee, a collaborator in Dundee. And using this information, we developed, so the first idea, trying to integrate these microbiomes. We tackled this with Jan had the idea to use a technique known as similarity network fusion, which I will briefly introduce now, in order to construct, to integrate these microbial data sets. So the network similarity fusion, as kind of prompted by the name, relies on deriving networks from these data sets. So the network inference in this case is based on looking for similarity between different patients. Basically, these are networks of patients. The nodes are individual patients, and the edges, the links between patients are inferred based on a similarity measure. So we use, in general, one can use different similarity measures, and in the tool that Giant developed and is available online, different similarity measures could be chosen. In our work, we decided to use a well-established measure for similarity from the ecological kind of studies literature known as Bray-Curtis similarity. Here, this slide presents the formula, and here is a worked example. Basically, the similarity between two patients is derived based on that expression here, involving the numerator involving the composition. A, B, and C here are microbes, and these are weighted by the total amount of microbes present in two patients here. This is a pairwise kind of measure. So using this Bray-Curtis similarity index, we can basically infer networks from our data sets, and for each individual microbial community, bacteria, fungal, and virome species. So after following this network inference for individual microbiomes, the next step is to look at how we can integrate them. So the similarity network fusion process allows us to achieve this. It proceeds in 2 steps. In the first step, basically decomposes each patient's networks into 2 subnetworks. One of them relates to capturing the overall network structure or similarity of a patient to all other patients. And here is the formula that describes how this is done with the work example. So this is denoted by P. And the second network, we just look at the similarity between patients in a neighborhood. Or in other words, we're looking at K most similar patients. And again, this is here denoted by S, and is given. The example is given here, a worked example of how this is derived. So these ideas, I should acknowledge, came from a publication by Bo Wang in Nature Methods 2014, where they have proposed this methodology in the context of genomic data. So we took this idea and we started by implementing this in the context of microbial data sets. And as I will show you, we needed to extend it and modified it slightly for the purposes of our analysis. So I just need to finish that. After decomposing these networks into the 2 subnetworks, then they get iteratively fused in a process that is best conceptualized as a diffusion of similarity information through common edges between the different patient similarity networks. So that's, after performing all this, what we get is a fused or integrated network of patients. Again, each node is a patient, and the edges now between the nodes contain information based on the 3 or more microbial data sets or microbiomes that have been fused. And this is here the mathematical representation of this process. The modification that we felt we need to implement relates to the fact that very often in this, for those of you who have experience with this metagenomics or targeted sequencing approaches, you would know that the quality of the species or taxa that get inferred after sequencing and aligning to available databases can be very unbalanced in terms of number of species or taxa that have been identified, and this is due to limitations in our knowledge in terms of the diversity of these kingdoms. So there are many species that have not been discovered yet, and the libraries are not perfect depending on the approach that is used to the bioinformatics analysis. So since the bacterial species have been studied for much longer period of time, usually the microbiomes derived based on bacterial composition are much richer compared in terms of number of species compared to the others. So, and that's here represented by these heat maps, where we have many more bacterial compared to to fungal or viral components in these sets. So we have introduced the so-called weighted similarity network fusion to take into account this unbalance in the number of species or taxa that we can work with. So we're basically weighting each of these measures that we derive based on the data sets, given the richness of the data set. So yeah, this is here represented. So we basically assign weight to each of these networks, and this is related to how many microbial components or taxa, or because they are usually taxa, they are not species. I know I'm using this interchangeably, but in our case, we use taxa rather than species. So this is an inherent problem in the quality of different biomes, and the weight is set relative to the number of microbes that are present at certain percentage. So these are all settings that one can choose when they are analyzing their data sets. And this is being implemented as a web tool, which is available online, and can perform this analysis of weighted similarity network fusion for any number of microbiome data sets. Using the user has the choice of different similarity measures, as you can see here, and then different weightings that can be chosen depending on the data set, on the specific data set. So what do we do with this and how we use it clinically? So far, it's really basically data analysis and developing some tools to handle these data sets. What we decided to do with the fused network is to, and that's kind of one natural step forward that one can do, given our hypothesis of stratification. So we looked for clusters and used spectral clustering in this case. This is just to remind everybody the data that we're dealing with this. So we, after performing weighted similarity network fusion, we proceeded with clustering. And this is basically, this clustering is totally data driven and doesn't assume anything in terms of properties of the patients. So basically we have the patient and the bronchiectasis is patient, the 217, and the similarity networks that we derived. So after performing spectral clustering, we find two clusters, and we ask the question, can they be mapped or related to clinical outcomes? So here you can see that these two clusters, the patients in these two clusters, quite readily map to several clinical outcomes, and importantly, exacerbations, which is the worsening of the patient condition. So we find that the patients in these two clusters are significantly different in terms of frequency of exacerbations and their history. So we call these clusters low exacerbation frequency and high exacerbation frequency clusters. So having identified these two clusters, we then ask the questions about, is there anything different in terms of how the various microbial species interact between each other as part of these clusters? And that is what we did this by using co-occurrence analysis, which is a quite well-established technique in this area. So the aim is to characterize microbial crosstalk in terms of the statistically significant microbial association networks representing possible interactions, which in our study we call interactome. We use the terminology of interactome. So again, there is a challenge that one needs to kind of face and take into account. The biggest issue, if I can call it, with these data sets is that they're compositional in nature, and that is due to the way they are derived and the bioinformatics pipelines that are used. So, and that in what is meant here by compositional, they are relative abundances. In other words, increase in one can naturally lead to decrease in another, leading in this sense to spurious correlations. So using correlative measures or association measures needs to be done with care. And in this specific study that was published in Nature Medicine, we use the approach proposed in this here paper that you can see. Although Giant, our student, is now exploring other approaches to compositional data analysis, the work is not published yet. That's why I'm not presenting it today. But basically, we employ an ensemble-based approach in order to deal with that compositionality and minimize these spurious correlations. So briefly, we calculate an association measure, and we use several different measures. We then create bootstrap distributions based on these measures, and then use permutation and renormalization to compare the bootstrap distribution and those obtained by reboot. So statistically, we compare this statistically, and based on the statistical difference, whether it's significant or not, we decide whether there is an edge or not, whether microbes are talking to each other or not. So this is the example of the co-occurrence networks that we find in these two different clusters. Basically, the most interesting result is that the microbial association network associated with the high exacerbation frequency cluster is less complex, and it's characterized by a greater proportion of negative interactions, microbial interactions. This is here kind of visualized on this slide, where you can see that there is many more red edges corresponding to interactions in the high exacerbation frequency cluster compared to the low one. We interpret this as an antagonistic interactome that is typical for the cluster of patients that are much more likely to have high exacerbation or worsening in their condition. Now that we have derived networks, we can also look at various graph theory measures, such as degree, stress centrality, betweenness centrality, that then can be associated with properties of microbial taxa. They can be characterized as busy, critical, or influential, given on these measures. And these are here the two networks that we consider: the low exacerbation frequency cluster and the high exacerbation frequency cluster. But moreover, we can just zoom into a specific pathogens that are known to be important for these patients and for this condition, and are targeted by antibiotics. And in this is just one example. In the paper, we have many more. If we look at the Pseudomonas, that's the target for antibiotics, and it's a pathogen that's implicated as important in bronchiectasis. What we find is that it interacts differently with Aspergillus, which is a fungal pathogen, again associated with this condition. We find that in the high exacerbation frequency cluster, the interaction between Pseudomonas and Aspergillus is positive, while in the low exacerbation frequency cluster, it is negative. I'll come back to this. This is still something that we are interested in exploring further. And in this paper, we just report this observation. We also looked at the small subset of this data that has a patient cohort that is followed over time, only of 17 patients, where we had information for their microbiomes at baseline, exacerbation, and post-exacerbation. So here, basically, what we have is these three different time points. And this is just to show that the composition of the taxa, in terms of well-established measures of diversity, is there is no significant difference between baseline, exacerbation, and post-exacerbation. However, similarly to what I showed you so far, and just confirming this in this small data set, during exacerbation, we find that the microbial association networks, or the interactomes of these patients, are dominated by negative interactions during exacerbation, which is not the case at baseline, where they're stable, and after treatment, usually with antibiotics. So we also looked at interactions that are conserved or that change for these different time points at baseline, exacerbation, and post-exacerbation. And we made several observations here summarized in this slide. I guess the most interesting one is that the interactions that change most and vary are the ones that involve pathogens well-established pathogens in this patient population, such as Pseudomonas, Haemophilus, and several others, Streptococcus, for example. So these are the ones that are associated with basically worsening of the condition and then improvement of this condition, again, consistent with what's known about these patients. We also did a little exercise here where we looked at whether we can predict the effect of antibiotic treatment using our methodology. So in a small subset, well, in 12 patients, 12 of those patients were treated with beta-lactam antibiotics. So what we did is we took their interactome and decreased the number of pathogens that are targeted with this group of antibiotics by certain percentage, in this case 75. Basically, this is to simulate the effect of the antibiotics, and then derived the network that then we compared with the observed one post-antibiotic treatment. And we could correctly predict 10 out of the 13 interacting taxa, which is a nice result given the complexity of the antibiotic treatment effects, as well as the complexity of these data sets. And very final for this paper and this study, that this example that I'm showing you, we also validated this approach in a cross-sectional cohort, completely independent cross-sectional cohort, which was sequenced using metagenomics approach. So here we have 166 patients from different sites around the globe, again, including Asia and Europe, Scotland and Italy. And we have their patients' samples, sputum, processed using metagenomics. So we take that metagenomics data and we apply this pipeline of analysis that I just described to you. Again, so here, in addition to the composition of the microbiomes, the taxa, we also have information about the gene functional pathways. Performing spectral clustering on these networks, we do find two clusters in the gene functional pathways, which are mapped well on exacerbation and this BSI score, which are clinical measures. So this is something which is gained additional information that we can gain from the fact that this is a metagenomic data set. So performing the same analysis, looking at the relative abundance of the bacteria, fungal, and viral microbiomes, followed by spectral clustering. Here, this graph actually summarizes what one would obtain, what kind of clusters one will get if it uses only the bacterial here in blue, the viral in red, or the microbiome in green. So the clusters, first, are very different in size, and secondly, don't map on any clinical outcomes in the same way as when you combine here in purple as the fused information from these three different networks. And these are the two clusters that we identify. Again, they map well on exacerbation, and looking at deriving their microbial association network for these two clusters, they are represented here, comparing them to the one that we obtained with the other data sets and the targeted sequencing approach. We find 89.9 common interactions in the high exacerbation frequency cluster, which is again a good sign that this methodology is robust and could be applied across different data sets and patient cohorts. So this all was published in Nature Medicine last year, and more recently we wrote this opinion article where we put forward the idea that a holistic approach to integrating multi-omics data and leveraging on their interrelationship is necessary in order to provide greater relevance and resolution for endophenotyping and ultimately clinical translation. So this is a new emerging area, I believe, in clinical respiratory medicine and care. Very quickly now, in the remaining 5 to 10 minutes, I will show you our latest work on this, applying these methodologies and extending them. This is a paper that came out a few weeks in the American Journal of Respiratory and Critical Care Medicine, where we are now looking at the crosstalk between the gut and the lung in bronchiectasis. So here we have a small number of patients, 57, but what is unique about these data sets is that we have concurrent samples from the bacterial and fungal microbiomes for these patients in the lung and in the gut. So they're collected simultaneously, the lung and the gut samples, and then they've been sequenced using targeted sequencing approach. So we have 228 microbiomes that we are working with from these patients. This is here just to show that we use the classical, we start always with the classical diversity analysis, and we find that there is a significantly decreased bacterial, but not fungal alpha diversity in the lung, which is probably, it's not surprising given that these are patients with bronchiectasis. So we also perform weighted similarity network fusion on these data sets and map them onto, this is just to indicate the robustness of the clusters that we obtain. So again, we identify 2 clusters and we can map them very nicely on clinical outcomes. So cluster 1 exhibits high risk of exacerbations, radiological and bronchiectasis severity. But what is really also interesting is that you cannot observe this if you look at the lung microbiomes only. In this case, there is not significant difference between the patients in the 2 clusters. And this is probably due to the fact that we are having much more sample here of patients, but also is indicative of the information that one can gain and the importance of the gut microbiome, suggestive of a crosstalk between those 2 as well in these patients. And I'm just going to show one more example of the kind of the analysis that we did. There are more results in the paper. But we also looked at a key pathogens implicated in bronchiectasis and their interactomes or microbial association networks in the lung and the gut, here as shown in this figure. And some of these things could be potentially used by clinicians to inform targeted antibiotic treatments or combination therapies for these patients. So far I talked to you about integrative analysis and microbial association networks. We have also started exploring the use of topological data analysis, which is relatively a new data analysis technique. And this is an example of the application of these ideas in a patient cohort with COPD. Looking here at the Aspergillus, which is a fungal pathogen allergens. So this is just a summary figure of the results. Using topological data analysis, one can derive, again, patient networks, which take into account the structure in the data. In this case, we have nodes that are patient groups rather than individual patients, and the links between nodes correspond to species in common or taxa in common that they share. So using this data set of Aspergillus allergens, we kind of, and performing topological data analysis, we identified 2 clusters. And as you can see here, we again map those on different clinical measures that are not used in deriving these networks. And we can nicely map those 2 clusters on different clinical outcomes, including exacerbations. So in summary, cluster 1, patients that are derived using topological data analysis are characterized by higher occurrence and frequent exacerbators, poor lung function, and worse prognosis. Cluster 2 of patient has higher symptom score, which is here shown in orange. But in addition to this, the topological data analysis identified further 2 subgroups that weren't apparent if you just perform traditional clustering on this data. And these are associated with specific allergens, again, potentially of clinical relevance in terms of therapy for these patients. Okay, I'm almost finished. The final few slides just show an example of how we're beginning to use mathematical modeling in these applications. So here the motivation came from that differential interaction between Pseudomonas and Aspergillus that we identified in our bronchiectasis cohort of low and high exacerbation frequency groups. So here we observe change of positive interaction to negative for the 2 different groups. So basically we're starting to model this using a classical, again, modeling approaches, Lotka-Volterra or pairwise interaction between microbial communities, which give us the possibility to take into account interactions between species as well as certain autoregulation of each of the communities. In this, so that's kind of schematic representation of the modeling. So what we're interested here is to vary these parameters and look for regimes that can lead to exchange in dominance of one species compared to the other. And here is an example. This is kind of a bifurcation analysis where we're looking at changing the interaction strength and identifying the parameter regimes where these interactions can lead to exchange in dominance, and very similarly, and the bifurcations that are related with this phenomena. Similarly for varying these so-called self-effects or autoregulation of each species within themselves. So the idea basically is that if we can manipulate these interactions in the lab using antibiotics or various other environmental factors, then we can control the system in terms of conditional co-exclusion or coexistence. And then that could be used especially if antibiotics are being used to change this relative composition of these pathogens that could then potentially inform future clinical applications. So with this, basically, I like to finish by acknowledging my clinical collaborator, Sanjay from NTU, who used this quote soon after we met, that there is only one way to see things until someone shows you how to look at them with different eyes. This has definitely been my experience in this project, and it's been very insightful. And I believe he's feeling exactly the same. Also, I would like to acknowledge our student, Giant, who has done fantastic and is now close to finishing his PhD. He's submitting his thesis in the spring next year. And the group of our collaborators at NTU, as well as in Italy and Scotland, who provided the data and some of the ideas that we then took forward and implemented computationally. So thank you very much for your attention, and I believe we have about 10 minutes for questions and discussion. Thank you very much.
Great, thank you very much. That was a really nice talk. Oh.
Shall I stop sharing? Sorry.
Maybe leave it up a second, just in case anyone has a question about referring to a particular slide. But then, yeah, we can. Bindi?
Yeah. Thank you very much for a really interesting talk into a completely new field that I have very little knowledge of. I was just trying to clarify to myself what you meant by positive and negative interactions when you got to that part. Do you mean so if one species increases, and I probably use the term species incorrectly, but if one thing increases, the other one decreases, or or have I misunderstood that?
No, you haven't. And that is actually a very common question. So that would happen because of the compositionality of these data sets. So we try to kind of minimize this specific reason for calling it positive and negative. But the way we think about this is antagonistic and synergistic interactions. So the way we infer them and cover them is based on whether the association measure is positive or negative. So they vary between -1 and 1. So if they're negative, we label this as antagonistic or red. And if they're between 0 and 1, we label them with green and as synergistic. So that's how we're interpreting and thinking about these things. Though, it needs to be taken with a pinch of salt because they are data-driven measures. They're just associations rather than causations. That's why we really want to go into a bit more mechanistic modeling approach. And I have, I'm not showing this here because we are in the process of analyzing it, but in Exeter, I have another collaborator who is a microbiologist, and they have started putting these pathogens together, growing them together and changing the conditions in terms of nutrients or antibiotics as well, exposure, and looking how they behave, what happens to them. And there is very interesting dynamics there. And we are in the process of fitting this data and trying to understand it better. In this situation, we would have much better handle on what is going on, but it's early days. So your question is very, very relevant. Thank you, Bindi.
Thank you.
Thanks. Himanshu, I think you are next.
Thanks. That was a fantastic talk. I have a couple of questions. One, just to follow up from Bindi. I noticed that in your exacerbation data, there was so much of negative interactions. What's happening there? And my second question is, what was your control for the 217 patient cluster? I mean, do we know what's the baseline microbiota look like for them or is like for them? And then you compare it with the patients and then you see, qualitative and quantitative differences.
Yeah, so again, very relevant questions. So the one with the negative interactions, the first question, it's basically the way again we interpret this is that the microbes are just suppressing each other, and that's kind of related to the decrease in the diversity, which is kind of known to be happening in some in some of these. In previous studies. What I'm trying to say is that having diverse microbiomes is actually healthy, and it helps these communities to thrive. While when they start to kind of kill each other or suppress each other, or not that, and when this diversity is disturbed in some way, that's then associated or leads to to worsening of a condition or to a pathology.
Are you suggesting that it's the microbes that are doing it to each other, or could there be external, human-related agents that are actually doing this? Maybe inflammatory cells or whatnot.
I think it's much more complex than it's not just the microbes that are doing it to each other. Definitely, external environment in this case, physiology, will be affecting them, but also they undergo treatment with antibiotics as well, and that would affect this
Makes sense.
As well. So I think it is very complex. That's why we haven't gone any further in terms of speculating on this, because it requires much more analysis and more data. So that brings me to your second question, where we only have a one time point. It's a cross-sectional kind of cohort. So the only information about what might be happening comes from the 17 patients where we had 3 different time points in terms of baseline, exacerbation, and post-exacerbation. And there we observed exactly the same. During exacerbation, we saw many, many more negative interactions. I went quite quickly over these slides. But yeah, again, this is only a very small one, and it's early days.
No. Yeah, you have to start somewhere. I think that it's fantastic, the data you've shown. Thank you so much.
Thank you.
Alex?
Thank you very much, and thank you for a lovely talk. And so my clinical area is cystic fibrosis, and we've had microbiome studies for a few years in CF, and what you report is well recognized, that patients with more severe disease have a much less diverse microbiome, fewer dominant species. And I suppose I'm particularly interested in how you sampled the patients and also how you sampled them during exacerbations, because patients who have had antibiotics recently, their different species will respond in a similar way. So there will be, the antibiotics are often quite broad spectrum. You may be hitting a number of different species. So they will go down, and it will look like there's a negative interaction perhaps on ones that are being released, but that's not due to those bugs interacting. That's just due to them responding to antibiotics, and particularly the case during exacerbations where they will definitely have had antibiotics. So two questions related to that. The cross-sectional 200 odd patients, how did you account for antibiotic use there, both chronic and acute? And the acute exacerbation patients, was that at the start of exacerbation before antibiotics, or was that while they're already been treated?
I really don't remember exactly, because that's usually, I would imagine though, I think they must have been all stable patients, because otherwise this whole thing won't make sense, right? And it's not going to pass reviews. But because what we do when we call them high frequency exacerbation or low frequency, we look at their records, like, because they're chronic patients, so they had some history of how many exacerbations they had in the past, and that's how we assigned these clusters. When we basically split it into groups. More than 3 is high, and less than 3 or something like that is low, and then we map onto these clusters. But I would imagine that their samples, and it's probably in the paper, must have been taken when they're like stable, because otherwise, as you say, the only ones that would have these other additional things that you were talking about are the longitudinal ones, where patients' data was collected when they actually have exacerbation. That had baseline. Yeah.
There's a cause and effect thing, because if they're frequent exacerbators, by definition, they've had a greater exposure to antibiotics. And some of them may in fact be on chronic antibiotics to prevent frequent exacerbations.
Yeah.
So I mean, it's fantastic work. I'm not trying to.
No, I know, and the antibiotic effects are definitely there. I'm sure they are. It's just very difficult to disentangle them with just a one-time point, basically. But I'm sure they are there. And Giant is analyzing some clinical trial data now, where, where we can see these we can make a bit more, because in clinical trials, everything is much controlled, and there are, much more longitudinal samples. I just not finished that work yet, but there we can say a bit more, something more specific about the antibiotic effects. Yeah, but you're absolutely right. Yes. So.
Great. And that was a good question. And Nazanin?
Yeah. Thank you very much for presenting this a lot of work, amazing work. I have just one question. So we have done some analysis in asthma using metagenomics data, and we saw that we have 2 clusters with different Shannon diversities. I'm just asking if you see there are like cell counts differences between your cluster. For example, we saw differences in neutrophils. This can be completely disease related, but that's just want to know if you see something.
Yeah, I'm not sure if they have looked at neutrophils, but actually that makes sense. What you're observing, I think. So so I don't have experience with neutrophils really, but I'm not sure how helpful this is. But it's you looked only at neutrophils or some other.
We looked at eosinophils and others, but the difference was in a neutrophil.
Right.
When we have a higher Shannon diversity, I think more neutrophils.
Yeah. I think that's kind of Yeah, it's probably, yeah, I think it's probably good to continue. So yeah, as I was trying to say in the beginning, there are studies, previous studies, and probably, again, people in the audience would know much better, that this kind of differences have been observed when you look at some communities. The focus of our work was to kind of combine those, really, to try to combine, to look at what we can find when we integrate the information.
Yeah, just one other question. You know, in the interactions, you showed a very nice kind of workflow, how you get there. My question is, how do you account for decreasing in diversity? So if, yeah, so one cluster has less diversity and probably, I don't know, more negative interactions, which can be kind of, how do you account for that? Or if you think this is important to be accounted for?
Is this something that you know beforehand, or you?
No, I don't know. No, it's just looking at your presentations.
So if it refers to the fact that sometimes you have much less information because of there is not enough libraries, or some species haven't been identified yet, or cannot be, or they're so little so that you can't really detect them in the sample. What we did, we applied weights to take into account that that's what we call weighted similarity network fusion. We basically, that's basically an unbalanced data set. So if we want to use them or to compare them, we need to take into account the fact that one of them is much less diverse, if I'm to use but this doesn't relate to alpha diversity or Shannon diversity or anything like that. This just purely relates to how many.
No, my question was about the interactions, when you have positive and negative interactions. And you had two clusters, and then one cluster has less Shannon diversity compared to other one. And then you show the interaction networks for each cluster. And then my question is that when you have less in one cluster with higher exacerbation, you have less of diversity, and then you expect to have less communications.
In our case, we did not find a difference in the diversity measures. Actually, they had the same, as I showed on the slide, they had the same diversity. So we don't really have that need to account for differences. Okay. The measures were not significantly different, basically, in our case.
Thank you.
Sorry. I guess we are out of time, right?
Yes, we are. Thank you very much, Krasi, for that talk, and thanks everyone for the great questions.